Rahman. Adeeb earned his doctoral degree in immunology from the University of Pennsylvania and postbacked both at UNC and here at Sinai. And it was here at Sinai where he began developing immune monitoring protocols while he was characterizing DC and macrophage compartments in patients with liver disease. He is currently an associate professor here at the Department of Genetics and Genomics, and is also the Director of Technology Development for our Human Immune Monitoring Core, where he oversees a wide range of advanced and high-dimensional technology platforms, including CITE-Off, single-cell-seq, and now CITE-Seq, which he'll be talking about today. So, Adeeb.
Thanks, Fiona. Hi again, everyone. All right. So, my title today is really on benchmarking CITE-Seq, which is this new and exciting technology that some of you have probably heard of. And the theme is really, perspectives from a converted skeptic. Because the idea sort of around the HIMC, as it's probably become clear for this meeting, is that our goal is really to take cutting-edge technology platforms together with sort of technical, scientific, and computational expertise and bring all of this to bear on our various patient cohorts that we have available to us at Sinai. And through many external academic industry collaborations. And so the cool technology, which we're hearing about today, is really one part of this equation. My focus is really on the single-cell approaches, flow and mass cytometry and single-cell sequencing. But a really critical part of being able to pair these technologies in sort of a meaningful clinical cohort or in the setting of large-scale translational research projects is really on balancing the innovation around these approaches with really making sure they're robust. And benchmarking them effectively to be confident in what we're seeing. And so as a sort of my role as Director of Technology Development largely involves bringing in some of these technologies, focusing on their utility, but as a corollary, one of my personality traits ends up being kind of useful here, which is that I'm deeply skeptical of everything. It's better to be a pessimist than an optimist, because either you're going to be pleasantly surprised or you're going to feel validated that you were right. So it's a good philosophy. But yeah, so my thought generally with most of these technologies is, we see something new, it looks cool, but does it actually work? So single-cell RNA sequencing, we're all familiar with this at this point. We've heard a lot of interesting stories about this already today. And so I think it's pretty apparent to most of us what the strengths of this approach are. It's a super high-dimensional technique. We can look at hundreds of thousands of genes per cell, and it's unbiased profiling, which is nice. So unlike typical flow cytometry or even mass cytometry approaches, you don't have to pre-decide on your antibody panels up front to decide what you're going to discover from your data set. You pretty much put a sample in, and you get out sort of what's there. And so this is a field that is developing incredibly rapidly. It's improving all the time, but there's still a number of significant weaknesses, particularly in considering its deployment over large-scale studies. And so one of the biggest impediments is a very high cost still. And there are these two elements of a cost per cell and a cost per sample overall. A cost per cell is often a more useful tool for companies that are promoting technologies. A cost per sample is the more relevant parameter for you as an investigator who wants to deploy these metrics in your studies. There is a relatively limited number of cells that are captured by most of these assays. So the 10x platform, which is the one that we tend to use at HIMC, typically, we're getting a maximum of about 10,000 cells per well in a typical experiment or per lane. And part of that limitation is due to this issue of doublets. So the nature of droplet-based microfluidics mean that the loading of beads and cells is somewhat stochastic. And so the more cells you try and force into one of these devices, the more likely you're going to end up with two cells that are aggregated with a single bead. And you can see this from the original sort of 10x publication, where as the number of cells goes up, the number of multiplets ends up going up as well. So this is part of what limits the number of cells that can actually be identified. Other major issues are high levels of gene dropout. So these are very sparse assays. You're typically only measuring a relatively small fraction of the total transcript in any given cell. And ultimately, you're also only profiling RNA transcript. And so obviously looking at many genes is useful, but genes aren't what are actually driving biology. Typically, those genes are encoding proteins, and those proteins are what are actually mediating what we care about. With these sort of known caveats to the approach, typically folks come to us and they're like, we want to do some single-cell stuff, and the questions are, so what are you trying to accomplish? Do you want to study proteins? Do you want to study lots of proteins, in which case you can use CITE-Off, or do you want to study RNA? And this has always kind of been a challenge in many studies, particularly in studies dealing with relatively rare clinical samples where you only have sufficient cells to run one of these assays. But then last year, there were these two papers that came out, one from the New York Genome Center, who are well represented here today, and another from Merck, both of which describe an approach of combining single-cell protein and RNA detection in a single assay. And so the rationale behind this is, when you're doing flow cytometry, you take an antibody, you couple it with a fluorochrome, you then use that fluorochrome as a surrogate for detection of the antigen you care about. When you're doing CITE-Off, you're coupling it to a rare earth metal and do the same thing. So here with CITE-Seq, we're coupling that antibody to essentially an oligonucleotide that includes a PCR handle, an antibody barcode that uniquely encodes what the identity of the antibody is. So this is, you could consider this to be your equivalent of FITC, for example, or PE. Except the nice thing with antibody barcode is with a sufficiently long string of potential oligonucleotides, you can exceed combinatorially the number of possible proteins in existence right now, or the number of antibodies in existence right now. So it's sort of an unlimited palette of potential labels. And then you have a poly-A tail on the end of this. And what this poly-A tail accomplishes is that it essentially turns this antibody into a synthetic polyadenylated mRNA. So you can take cells, label them with a cocktail of these antibodies, put them through any conventional single-cell sequencing platform, such as the 10x platform. When that cell is encapsulated in a droplet with an antibody capture bead, the cell lyses, you now have mRNAs or poly-A labeled mRNAs from the cell itself, so the native mRNA from that cell, and the synthetic pool of polyadenylated mRNAs derived from those antibodies. Essentially, these two libraries can be processed in parallel. So from a single cell level, you can now get both the RNA as well as the antibody profiling. So when we saw this technique, as is usually my inclination, my first inclination was, this is super cool. My second was, clearly this doesn't work. And so I ended up contacting the guys at New York Genome Center. We got a little grant together, partly to further benchmark technology and deploy it in a larger set of settings. So here's an example of sort of what we're trying to accomplish. So one also a note about how we're going about doing this is that for those of you who are accustomed to dealing with single-cell seq data, there's a lot of sort of data transformation gymnastics that people tend to do to be able to visualize these sorts of data sets. So people will z-score things, and they will visualize them in t-SNE plots and UMAP plots, and these things are great for making pretty figures to go in papers. And they're also very good for being able to visualize broad structure and gain insights from your data. But one of the corollaries to these approaches is they dramatically mask the underlying data quality. You can take really crappy CITE-Off data and make a beautiful looking t-SNE plot out of it. And it's very unclear how some of these things sometimes go together. So in sort of benchmarking this approach, which is kind of a radically new way of detecting proteins, we wanted to compare it to sort of traditional ways of measuring proteins on single cells, so flow cytometry and CITE-Off, which is pretty established in my mind at this point. So here's how, as a cytometrist, I would look at two proteins by flow cytometry. So here we have CD4, we have CD3. We can gate on a population of CD4 T cells on the basis of this data. Pretty standard approach. So when we run CITE-Seq and we deploy the CITE-Seq count script, is initially developed by Marlon Stokias. This is what the initial raw CITE-Seq data looks like, straight out of the count script. Didn't look so great initially, but don't be alarmed. Very easy fix. So the first thing we do is we just do an arcCNH transformation of the data. So that's exactly what we do for CITE-Off. So the idea here was, can we take the same data analysis or data transformation pipeline we deploy for CITE-Off and apply it for CITE-Seq? So step one, arcCNH transform. Things are already looking a lot better. You'll notice that there's a sort of picket fencing effect in the data here, and that's largely it's just a measurement effect. It's because the data is being measured in integer format. And the reason you typically don't see this in flow cytometry is because the measurement flow cytometry is imprecise. So there's a lot more noise that gets added to the data as a result of compensation and measurement artifacts and things. So all of your integer data essentially becomes a little bit more imprecise. The reason you don't see this in CITE-Off data is because we compensate for it by adding a little bit of random noise to the data. So essentially every integer value is shifted either plus or minus 0.5. So it's a really small effect. It's enough to essentially randomize the data without changing the overall measurement of any given parameter. So we apply the same trick here. We add a little bit of random noise, so every value is slightly changed. And you can see this has sort of no appreciable effect on positive populations, but it does smooth out this visualization in the lower ranges. And having done this, we can now gate in the data just as we would by CITE-Off. So things are looking pretty good. Of course, one of the points of this exercise, we now want to enumerate the frequency of that population we've identified as a function of all cells. And when you do that, things, again, don't look so good. So here we have 33% of the population looks like T cells, and only 0.3 in this case are CD4 cells in the CITE-Seq. But this is, again, a very solvable problem. It's just one of the denominator. So the issue is, what do you call a cell when you're doing these approaches, right? Because this is a microscopy, and all of these things, you have some sort of digital form of data, and you need some way of deciding what is a cell versus what is not a cell. And so the way we typically deal with this in CITE-Off is we use nucleic acid intercalators, so that essentially non-specifically label nucleic acids or DNA in cells, and we say that anything that is positive for DNA is a cell, whereas stuff that is sort of not negative for DNA ends up being kind of a smear, and we gate that out as debris. So there's sort of a similar approach here for single-cell seq, where there's a vast excess of capture beads in any given experiment, and only a small subset of those beads have actually bound to a cell. And so this is the standard Cell Ranger pipeline, which uses a UMI threshold, and it says anything that has a detectable amount of RNA is a cell, and anything that has a very low amount of RNA is likely an empty bead or debris, or bead binding to sort of free debris. Again, as a cytometrist, the way I prefer to, I find these plots very sort of counterintuitive or hard to think about. So you can visualize the data in kind of a similar way to this, which is the gene expression UMI on the x-axis, the unique gene count on the y-axis, and this ends up forming a plot that's pretty similar to what we're kind of accustomed to looking at. We can use this to threshold on what's a cell. So that's one approach for CITE-Seq. We could take the gene expression data, we can threshold on that and say that based on gene expression, this is what we're calling a cell, and we can port those numbers back to CITE-Seq. But an alternative strategy is to try and have a method internal to CITE-Seq itself to tell us what a cell is. So instead of using DNA, one thing we can do is just use some sort of ubiquitous marker on the cell. So if you're studying immune cells, for example, something like CD45 is a nice approach. But if you're going to do that, there's another variation on this that you can do, which is called sort of sample hashing. So you heard about in Alad's opening talk today about cell barcoding, which is what we do for CITE-Off. So barcoding is something we do routinely in all of our CITE-Off experiments. It's a great way to be able to label samples and pool them together. And so we thought, why don't we do exactly the same approach for CITE-Seq? And Marlon actually very early on corrected me when I said, "Oh, we should just do cell barcode." And he's like, "Well, there are already cell barcodes in CITE-Seq. You can't do that." So that's the term cell hashing, which was coined by New York Genome Center, is I think a much more apt term in this case. And so the idea here again is you have various samples. You label those samples with antibodies targeting some sort of ubiquitous protein on the surface of those cells. You can now pool all those samples together into a single sample, load that into your instrument, and you can computationally deconvolve these cells back into their original source samples. And a nice sort of advantage of this approach is it also helps you filter out cross-sample doublets, because you know that a cell can either be red, green, blue, or one of these. So any cell that happens to have expression of more than one hashtag can be identified as being unambiguously a doublet, and we can remove that from our data set. And so this is actually a huge benefit to the approach, because as we talked about before, doublets are a major problem in single-cell sequencing. And the more hashtags you include in an experiment, the greater the number of doublets that are stochastically going to happen across samples and can be filtered. So by pooling 10 samples together, we can essentially eliminate 90% of the doublets from a given experiment, which also has the added benefit that we can load more cells into these assays than would otherwise be possible. And so this strategy does require some sort of ubiquitous protein on the surface of the cells. So we tested a bunch, and actually the legend screen experiment that Alad had showed you earlier was kind of a nice way that we screened for markers that were ubiquitously expressed in all these cells. And based on the CITE-Off work we were doing already, we settled on a scheme of using beta-2M and CD298. Both of these are ubiquitous proteins, but also importantly, we'd shown through some CITE-Off experiments that neither of them induce significant signaling. So you can bind antibodies on the surface of the cell, and it doesn't seem to do very much to those cells. So you're not inducing a transcriptional change by virtue of barcoding. And conveniently, I guess it's a nice validation that you get things right. A paper was recently published from Stanford that showed the same approach for CITE-Off, and I'm pretty sure BioLegend is using the same combination of antibodies in their commercial hashing reagents now. So that seems like a pretty good strategy. So having deployed this approach, we now have a new way of being able to identify cells in single-cell seq. And so what you're looking at here is on the x-axis, we have expression of the maximal primary hashtag signal. So essentially, what is the highest intensity for any given cell? So the idea is that cells that have been bound by an antibody are going to be positive. Cells that do not bind an antibody are going to be negative. And so this is a good way to be able to identify cells versus debris. But now we have a second metric, which is what is the intensity of the second highest hashtag? Because keep in mind, as I showed you before, cells should only have one and one only. If a cell has two hashtags, it's going to be a doublet. And so here we can very easily identify our doublet population by doing this. And one kind of nice insight that comes out of this is if you now plot gene expression UMIs against this metric, you can see that it's been proposed for single-cell seq before that maybe you can filter out doublets from your data by eliminating cells that have relatively high levels of UMIs. And while that is sort of true, the doublets do in fact, on average, have higher UMIs than the singlets. They're clearly overlapping distributions, so there's no way you can threshold and accomplish this without having the hashtags in the experiment. So it's a really nice way to clean up data that wouldn't be possible otherwise. And so having done this, we can now take that singlet population, we can deconvolute the individual samples that went into it. So this is, for example, our 10 different sample hashes. We can identify our 10 different samples that were encoded with those hashes and then break down the relative frequency of those and now analyze all those samples sort of individually. So, so now that we have this way of filtering data, this was the raw data I showed you before with CITE-Seq on the top, CITE-Seq on the bottom. But now after filtering on single cell events, we end up with a frequency that looks much more comparable. So this is pretty reassuring. So one thing I also want to note is that this approach of filtering your data based on a hashtag works pretty well with traditional sort of UMI-based filtering, at least if you're manually thresholding. I should note that Cell Ranger, having sort of started to visualize several data sets in this way, it's kind of become apparent that where Cell Ranger draws its default threshold is often kind of pretty wacky. It'll just like randomly cut this distribution somewhere in like the middle third and things like that. But by plotting the data in this way, usually you see a pretty nice inflection point in 2 dimensions where you can filter cells on the basis of your gene expression, you can filter cells on the basis of the hashtag, and they align pretty well. But a lot of our interest was not working in blood, it was work with tissue samples, and we found that this did not work quite as well when we started getting into tissues. And as we dug into this deeper, we actually found one case where it was most dramatic, was if we're actually dealing with RBC depleted whole blood. So this is a PBMC, so cells that have undergone a phycol enrichment. This is just total whole blood where we've removed our blood cells. And you see this huge population of cells down here that are clearly positive for our hashtags, so they're cells as identified by protein, and they are not cells as identified by gene expression. This is pretty strange. And you can see this big discrepancy here. And so to dig into what was going on here, we took the data as filtered by the cell hashtags, and we just clustered it based on the antibodies. And using this, we can identify all these major immune subsets. So most of the canonical things you'd expect to see in whole blood. But now if we take the same TST map and we overlay the relative gene expression on top of it, you will notice something quite dramatic, which is that this huge island of cells over here, which are the neutrophils, are pretty much devoid of UMI transcripts. Another way to look at this data is just to do this, to take each of these different subsets and rank them based on their relative gene expression. And so you can see that most of the cells that we're comfortable looking at, T cells and monocytes and all those guys, have detectable UMIs, whereas all of these granulocyte populations, most notably neutrophils and eosinophils, really don't. And so when you do a basic UMI-based thresholding, you essentially cut these out of your experiment. And if any of you were paying really careful attention during Jerome's talk, when he showed this like beautiful sort of alignment between single-cell seq and CITE-Off, there was a little line of cells that was all the way on the axis that were positive by CITE-Off and not there by single-cell seq, and those were the neutrophils in those biopsies. And so this is kind of a nice sort of example of what sort of a discrepancy between these 2 approaches. So one question, of course, is what's going on here? Is it true that neutrophils just don't have any transcript? Because that would be kind of surprising. Another thing that's been mentioned sometimes is, well, they're just dead, right? Neutrophils don't make it through encapsulation, so they're just not, they've died and somehow lost all of their RNA along the way. So to try and get at this question, we did a little sort of the analysis trick. So the nice thing here, keep in mind that we have these cell hashes, so we can unambiguously identify doublets in singlet samples. By doing this, we can identify T cells, let's say on the basis of CD3, neutrophils on the basis of CD66B. And now looking within the doublet population, we can specifically identify T cell and neutrophil doublets versus T cell doublets with other kinds of cells that are not neutrophils. And if we take this and now plot our relative gene expression, you can see that here are T cells, here are neutrophils. T cells have a lot of UMIs, neutrophils have no UMIs. When you have a T cell that forms a doublet with something else, you have extra UMIs, as we've seen before. But when a T cell forms a doublet with a neutrophil, you actually lose expression from these cells. What this is really suggesting is these neutrophils are actually chewing up transcripts. They're actually loaded probably with RNases, and when they end up in these bubbles and they lyse, you get release of all these RNases, and it depletes the transcript from these cells. So that was kind of interesting. And we thought, well, if that's what's happening, perhaps one way that we can mitigate this is to try and load a 10x lane with neutrophils in the presence of additional RNase inhibitors. So clearly the 10x reagents contain a bunch of RNase inhibitors, but maybe more is better. So we tried doing that, and the bad news, unfortunately, didn't do anything. And so we thought of doing this in a more rational way and trying to figure out exactly what the neutrophil RNase profile looks like, but I didn't have any money to do it, and so we thought we'd just go the brute force method and just throw in the most potent RNase inhibitor cocktail we could. But doing this, you can really see that the neutrophils remained largely low for gene UMIs. There's maybe a tiny increase, but not to the point of being really noticeable. But one other kind of nice thing that came out of this experiment, because it's something that I think has been, I've heard proposed at a few meetings, maybe you can just boost your general gene expression signal by having more RNase inhibitors in your cocktail and being able to recover some of that. And that at least does not appear to be the case in this experiment, because looking at the non-neutrophil cells, they cluster essentially identically with and without the RNase inhibitors, and there's no sort of appreciable increase in the overall unique gene counts across any of these subsets. So if you're thinking of doing this, save your money and don't bother. But so the bad news is that we aren't able to recover these missing transcripts in neutrophils. So that's the downside. So they kind of remain ghosts in the single-cell seq assay. But the nice thing is we actually can account for their presence. And this has actually been one of the things that was a very frustrating aspect of sort of running single-cell seq as a service for a while, was that we'd have people who'd come with a sample, they'd load it, and they say, we've counted your sample like 15 times using 5 different methods, we've loaded 7,000 cells, and now all of a sudden we only get back 2,000 cells. And they're like, you guys suck, we hate you, we don't want to pay your money anymore. And we'd be like, I'm pretty sure we did it right. And this is, I think, what's been going on. Like, where there's minimal contamination in these granulocytes is a way of essentially cells that look like cells when you load, but just don't appear as cells when you actually do the assay. Yes? We should do an entire procode library of every possible transcript and then see what happens. Yeah, if someone wants to pay me to do that, I would love to. But as of now, no one's probably going to do that. But if you want to, come talk to me later. Right, but the one nice thing about this though is even though these neutrophils kind of remain again not really analyzable by transcript, it does allow us to account for their presence. And so here you can see that same example of that whole blood data set. And if we filter it by gene expression, we ran parallel aliquots of the same sample by CITE-Off and CITE-Seq. The relative frequency of those cells is totally divergent, right? Because the neutrophils are essentially present in one assay, they're not present in the other, and so everything else as a corollary that ends up becoming a little bit off. But now by filtering based on hashtags, we end up completely aligning these relative frequencies from the samples. Another nice thing here is, it's all the way. This was only about 7,000 cells overall from a whole blood sample, and even from those 7,000 cells down to PDCs, which were only about 0.2% of the sample here, we're getting very good alignment between the two approaches. So we've kind of expanded this out to much larger panels now. We've kind of really gone full force with this, and so here's just some more examples of some data showing you CITE-Seq on the top here, CITE-Off at the bottom, and I've just manually gated this data as a cytometrist probably would. And you can see across the board, these profiles look very, very similar. The analogous populations can be gated very easily. The relative frequencies of these gated populations remain very similar, all the way down to pretty rare subsets like plasmablasts and transitional B cells and things like that. So this was really, really encouraging. We've also run automated clustering on this. This is using the Astrolabe platform, which you'd heard about earlier on. I should mention this is not an out-of-the-box thing. We have to have a lot sort of hack the platform a bit to accommodate for this. But essentially, this is CITE-Off data and CITE-Seq data analyzed with a matched panel of 37 antibodies. We're able to use flow-sum clustering to identify analogous populations across both methods, and the alignment between these populations, again, is very good. So based on this, we're pretty confident that the two approaches are working very well together. We wanted to go even further here and really try and get into benchmarking the sensitivity and the linearity of measurement across these two methods. And so the trick we did here was something we'd actually done early on, when we were benchmarking the CITE-Off several years ago, which is try and use fluorochromes as a model antigen, where we can do sort of controlling the antigen density on any given cell on the basis of fluorochromes. So here, this is an example of PBMCs where we stain the sample with increasing amounts of PE-CD64 antibodies, which is a monocyte-specific marker. And you can see that as the antibodies go up, you now have increasing amounts of PE on those samples. We're now taking these same samples and we're staining them with an anti-PE antibody, either conjugated to metal, analyzed by CITE-Off, or conjugated to an oligo and analyzed by CITE-Seq. And just visually, already you can see that this looks pretty good. Things align pretty well. To make this even more quantitative, we use these absolute quantification beads, allow us to convert this PE fluorescence intensity into an absolute count of the number of molecules per cell. And by doing this, we can now plot essentially the measurements for the number of molecules per cell versus the intensity is pretty much completely linear across both of these methods, across a pretty nice dynamic range. It is true that at the very low end, things do taper off with both methods. Kind of a more meaningful way also of staining intensity is one thing, but the thing we often care about as as immunologists identifying these populations is, can you define positive from negative? That's like the real question. So if you have a cell that is known positive for a marker and a cell that is known negative for a marker, can you resolve that well? And so that's this is a metric that we came up with a couple of years ago called the average overlap frequency. Essentially, it's a measure of how many cells in a positive population overlap with those in a defined negative population. So one is is bad. Essentially, 0 is great. It means there's 0 overlap. 1 is bad. It means there's complete overlap. And you can see that as the number of PE molecules drops, the relative overlap goes up. In this particular experiment, CITE-Seq was a little bit worse than CITE-OFF. We've had other experiments where the ratio was flipped, but in general, they're pretty comparable. But another important aspect of this, though, is that with the CITE-OFF, our detection efficiency is essentially fixed. Like the machine pretty much has a sensitivity. It's not like a flow cytometer where you can like tune your PMTs and tweak all that stuff. Pretty much like what you have is what you have. The only thing you can really do is optimize your instrument. The difference for CITE-Seq, though, is this is a highly tunable variable because sequencing depth essentially is what regulates your intensity. So the more deeply you sequence your sample, the greater your staining intensity is going to be. And so you can pretty much drive this up. Eventually, you will reach saturation. So there will be a saturation point where you've gotten all the possible reads. That seems like pretty infeasible to reach that with anyone's typical budget, and it also seems largely unnecessary. Because there's sort of diminishing returns to having greater intensity, because at some point, if your two populations are well separated, why separate them even further? So a downside to this, though, it does mean that if you are trying to compare data across experiments, you have to be very careful about trying to match your reads if you're trying to do anything quantitative. But an even sort of more complicated aspect, which is sort of apparent from this, but I think bears recognition, is that not only your sequencing depth, but the complexity of your panel is also a big driver of this. Because unlike CITE-OFF or flow cytometry, where your measurements of different markers are essentially independent, you have one detector for marker X and one detector for marker Y. In the case of CITE-Seq, you essentially have one pie of detection of all your antibodies, and you're slicing that pie up between all the possible things you want to measure. So the more cuts you make in that pie, the more complex your panel becomes. That's effectively has the same effect as reducing your reads to any individual marker. And you can see that here. This is the same number of total reads across these two experiments, and the same number of total APC molecules, the same amount of antigen. But in the context of a more complex panel, you effectively driven down your staining intensity relative to a simpler panel. So all this to say that I think CITE-Seq within any given experiment is beautifully quantitative. You can see how clearly linear all these measurements are. But when you're comparing across experiments, doing any sort of absolute quantification becomes a pretty fraught exercise because there's so many of these aspects of complexity that go into it. So I think this is a great approach for being able to define subsets on the basis of differential expression of markers. It's probably not a great approach if you want to do an absolute quantification of PD-1 expression between 15 different patient samples, for example, unless you barcode them all together and you run them one time. Cool. So hopefully all this has convinced you that antibody detection by CITE-Seq is largely comparable to CITE-OFF, which is great. But of course, the thing that most people want to do is also look at gene expression. This is a single-cell sequencing, after all. And so one of the key questions also was, does having all these antibodies in the cell have some effect on our gene expression measurements? And the good news is it does not seem to, in our hands, at least. So here is an unstained sample, for example, clustered based on gene expression. This is that same sample now stained with antibodies. You can see the overall antibody counts are much higher here, but there's no overall change in the distribution of cell populations. There's no overall change in the overall distribution of unique gene counts. This is an experiment we actually did a titration showing increased amounts of antibodies going into the sample. And as the antibody amount increases, there's really no obvious change in the median genes or the median unique genes or the median UMIs across these cells. So we're pretty confident that we're not sort of destroying all of our conventional single-cell seq by doing this. And I think one of the things that really comes out of this, so one of the points to note is that I think when some of the folks first started thinking about this, they were like, oh, okay, well, it's nice that you're looking at protein. It's a nice add-on to RNA, but we have hundreds of genes. So what are your 10 proteins going to do for this experiment? And I think one of the things that's really come out of CITE-Seq is it really highlights how much better protein detection is in many contexts than transcript detection. So I think we all know that dropout is a problem in single-cell seq, and I think this is a really nice way to illustrate that. So here you have CD14 and CD8 antibody, which we're using to define monocytes and CD8 T cells respectively. And now if you plot the CD8 gene versus the CD8 antibody, you can see that they are correlated, so that's good. Generally speaking, there are most CD8 T cells have CD8 transcript, but one thing I want you to note is just the magnitude of these axes, right? So this is a 4-log axis that we typically use in flow cytometry. So these measurements are in the hundreds to thousands range. These are single-digit measurements. So with monocytes, it's maybe a little bit better, but still you're typically in the single-digit range. The other thing that's important to note is this first column over here. So these are cells that have detectable levels, and not just detectable, but they're being defined on the basis of a canonical antibody marker. These are gated CD8 T cells. We're calling them CD8 T cells because they have CD8. And there's a very large proportion of those cells that have no detectable transcript for that gene. likewise with monocytes. So we went through this exercise for 20 canonical genes or 20 canonical antibodies in our panel. That's what you see here. So this is for all these 20 antibodies that are just ranked. In orange, you have cells that were positive for the protein and were also positive for the gene. In blue, you have cells that were positive for the protein and had no detectable transcript. And you can see it clearly varies depending on the gene. Like some genes, so CD161 is very robustly detected at the transcript level. Other genes, many of which are pretty canonical, like CD4, 90% of CD4 T cells have no detectable CD4 transcript in this experiment. I will note that we didn't sequence this to saturation. Obviously, the deeper you sequence, you can mitigate this to some extent. We already have about 10 times as many reads devoted to our genes as we have our antibodies. I think this is about 30,000 reads per cell devoted to our genes, and only about 4,000 reads per cell devoted to our antibodies. And so, one thing you could do is, just keep sequencing your gene deeper and deeper, and maybe you'll be able to recover some of this. I'm sure it's never going to get anywhere close to 100%, but it's a much cheaper approach to be able to compensate for this poor gene expression by simply having the antibodies. Now, the other complaint about this, of course, is no one uses single-cell seq to look at individual gene measures, or at least no one should be using single-cell seq to be looking at individual gene measurements. If you're doing that, stop. Bad. It's wrong. Don't do it. Another also related message out of that is that single-cell seq, while it is a single-cell approach, you shouldn't really be thinking about it as a single-cell analysis. A single cell in single-cell seq is pretty much meaningless. Like, all the measurements you need to do if you want to learn something about them have to be at the population level, which is why we look at clusters, right? So it's important to know about consistency of a cluster, but really it's averaging across multiple cells to compensate for some of these issues. But that being said, of course, why would you only use one gene to look at something with single-cell seq? You want to use lots and lots of genes. But even if you do that, so this is an example of PBMCs. They're clustered, there's a t-SNE based on the top 200 differentially expressed genes in the sample. And you guys have probably seen plots like this before. You can see that it does resolve things pretty well. We see a nice separation of sort of CD14 monocytes, CD16 monocytes, T cells, B cells. But you can see that within our B cells over here, we have pretty poor resolution of naive and memory B cells. Within our T cell compartment, the CD4s and CD8s are largely kind of commingled. The NK cells are kind of pretty hard to separate from the CD8 T cells. You've got this kind of cluster of apparent cells here, but really they're just characterized by having low UMIs. So probably somewhat dying cells. So if you take the same data set and now you analyze it with just 20 antibodies, so we're using only 1/10 the number of antibodies, we can, some things are better, some things are probably worse. But largely speaking here, for these canonical known cell types, this very small number of antibodies has given us sort of much better resolution of our CD8s or CD4s or NKs. We can resolve our naive and memory B cells. So the addition of a few antibodies can really make a dramatic impact to having all of these different genes. And this is even more key when you start getting into sort of finer grained subsets. So here, for example, we're looking specifically at CD8 T cells. And so these are just 6 antibodies looking at CD8 T cells, but they're antibodies that have a lot of historical precedent, right? So this is how we define all our canonical T cell subsets. We have our naive T cells, our high for 45 and 62L. We've got our central memories that have lost 45 and maintain 62L. We've got all our various effector memories, which can be further stratified based on CD27, 28. We've got some mate cells. And so, there's tons so one note about this is these may all be somewhat transitionary states, but there's a lot of historical precedent to know that these are functionally distinct subsets, and they are along a spectrum, but there are functional differences between EM1s and EM4s and EM3s, for example. And if you take the same data, you cluster it based on the top 200 genes within the CD8 T cell compartment. So we're focusing on the CD8 T cell compartment. We do see nice stratification between the naives and sort of the non-naives. The mates actually separate pretty well as well, because largely because CD161 is actually very highly expressed at the transcript level. But there's pretty much a commingled expression of all of the other different clusters. It's very hard to resolve by itself. And so the nice thing about CITE-Seq is it lets us now take kind of our historical understanding of the immune system, of which there is a lot, and kind of overlay that on top of sort of this unknown gene-based clustering that's kind of the new frontier. But not having this can make it a little bit harder, I think, to make sense of some of this. And Nick's talk later today, I think, is going to talk about some of the visualization approaches, analysis approaches to accomplish this a bit more elegantly. All right, so I'm going to end there by concluding that hopefully I've shown you the detection of these oligo-labeled nucleotides, oligonucleotide labeled antibodies by single-cell seq is actually very comparable to flow cytometry and CyTOF. The antibody-based hashing is really a great method that I think allows for more robust cell identification and double exclusion, and I think it's really improves data quality. The antibody staining doesn't really negatively impact our conventional single-cell seq. And the antibody is providing valuable information on top of single-cell-seq. So despite my initial skepticism, at this point I would say, why would you even do single-cell RNA-seq when you could instead do CITE-Seq? All right, so I'm going to end there by acknowledging several people, in particular Laura Walker, who besides being, turns out, surprisingly, an event organizer extraordinaire, is also an exceptional single-cell-seq technician and is really responsible for a lot of the bench work to get this stuff done, together with Chrissy Chang from Miriam's lab. Melanie and Nick have been indispensable for all of our data processing, as well as the rest of the HIMC team. The folks from New York Genome Center, so Marlon and Peter, who really were instrumental in getting this project off the ground. And the folks from BioLegend, who helped a lot with transitioning some of these from our initial sort of homebrew antibodies into sort of commercial reagents that I think are giving us more sort of high fidelity data at this stage, and a little grant for funding for this project. I'm going to end with also one last final plug, which is if you enjoyed this talk or any of the other talks today and you want to work on this, come talk to me because we're hiring. So we've done a little bit. It was sort of a hacky way of doing it in that we used our fluorochrome conjugated antibodies to do an anti-tetramer. So it was an APC conjugated tetramer, which we detected with the anti-APC antibody. I think the next talk will probably give us a much more elegant aspect of this, I think, unless I'm lying. You're not going to talk about tetramer? Okay. Maybe the following talk will give us